of the greatest mysteries on Earth is the origin of life. Where did it come from? One theory supported by the Miller-Urey experiment explains how early Earth conditions facilitated the synthesis of organic molecules, which later led to formation of cells. Deep-sea vents, delivery of life forms via meteoroids, and special creation also offer possible mechanisms for this event, and they can help to explain how we as humans are present on Earth today. This is where theory of knowledge can help us. TOK allows us to understand how we know what we know and how knowledge is created in the different areas of knowledge. As you should know, biology is part of an area of knowledge, the natural sciences. We can then use our ways of knowing to ask how do we know and how do I know? The ways of knowing include language, sense perception, emotion, reason, imagination, faith, intuition, and memory. Through TOK, we can begin to investigate the dilemma around the origin of life. The way knowledge is created in the natural sciences is through the scientific method. We must then use sense perception to collect evidence in response to a hypothesis. This evidence, through reasoning, is used to develop theories, generalize from data to form laws, and propose hypotheses. These theories and hypotheses are used to make predictions that can be tested. In this way, theories can be supported or opposed and can be modified or replaced by new theories. This automatically rules out special creation from what we can study in the natural sciences because it relies heavily on faith, which cannot be verified through sense perception nor reason. But that isn't to say your faith is not valid. We just won't go there. One part of the cell theory is that cells arise from only pre-existing cells. The challenge with this statement is that cells had to somehow be present on Earth to lead to the evolution of life into the many diverse forms it appears as today. How do we know when scientific conclusions are justified? Again, it is through collecting evidence to verify our hypotheses and theories. While it may not be possible in our lifetime to definitively verify how life originated on the third rock from the sun, we can test the general principles that underlie the natural world. For example, our third part of the cell theory. Before we were able to take for granted that cells are produced by existing cells, there was a dominant theory called spontaneous generation. This theory states that living things can appear without stimulus or external causes from non-living matter. Recall that scientific discoveries are limited to the tools and apparatuses of their time. For the theory of spontaneous generation, we can thank Aristotle. Aristotle, the Greek philosopher and scientist, is credited with many things, including the theory of spontaneous generation. He made very basic and simple observations of many invertebrates. For example, he observed shelled mollusks appearing and growing in their aquatic environments. He argued that non-living matter possessed some sort of vital force, and you might be aware of it more or less. The original elements: water, earth, air, fire, and a vital force. The proportions of each in an object mattered, since living things required specific proportions. And since environments vary in composition, Aristotle also observed that these living things are products of the abiotic factors of the environment. Clams are found in sand, oysters in mud, and barnacles on rocks. Discoveries are products of their time, and Aristotle's conclusions were widely accepted for about 2,000 more years. In the meantime, before we get to our next old dead white guy, spontaneous generation was adopted by Christianity. In the Christian Bible, there are a lot of allusions: the breath of life, or the generation of rats in Egypt due to overflowing rivers, or the beginning of Genesis, and so many others. This theory and its relationship to religion implored scientists to be careful in collecting evidence to not only verify or falsify spontaneous generation, but also avoid being labeled as heretic. This was the delicate game Italian physician, naturalist, biologist, and poet Francisco Redi played. The basic premise of his apparatus was he placed rotting food in containers and either covered or uncovered them. If you research more information about him, you will find that his experimental design was not really that great, but he continued extensions of his original experiment in attempts to develop a well-rounded principle that underlies the natural world. His results were consistent. Covered meat did not spawn maggots or flies, but uncovered meat did. Flesh exposed to dead flies did not spawn maggots and flies, but ones to live flies did. His results were carefully phrased, but more importantly began the process of building evidence to falsify spontaneous generation. Omne vivum ex vivo. Pier Antonio Micheli, Italian priest and botanist, continued what Redi began. He worked primarily with fungi and famously became the first person to culture fungi from spores. This is actually what helped falsify spontaneous generation. Fungi could only develop into the same species that was placed on the fruit. All life comes from life, and like begets like, also non-threatening to the church. By the time English biologist John Needham was experimenting with the theory, it was known that boiling liquids would kill microbial life. Needham boiled broth, cooled it in open containers, and then covered them for several days. The turbidity became obscured, showing that bacteria could spontaneously generate, and provided support for the later review of his literature showed that the duration of boiling was insufficient to kill microbes. There were many others involved in the contributions to verifying the theory, both in support and opposition, but it was generally agreed for a conclusion that if air was allowed to come in contact with the specimen, spontaneous generation could occur. All of this debate would come to an end with Louis Pasteur. He wanted to verify the principle that air exposure could spontaneously generate life from dead and non-living matter. So Pasteur designed an experiment to involve broth that was boiled with sufficient time to kill any microbes, and did it in swan neck flasks. This would allow the broth to be in contact with air, but the design of these flasks drastically reduced the movement of air in the flask. His control group involved breaking the neck of the flask off, whereas the experimental was left intact. The control group grew microbes, but the experimental did not. This falsified the principle that air exposure could spurt life. In a second experimental group, he tipped the flask so that the broth would come in contact with the neck. This group did grow microbes. This led him to developing the germ theory, which then led to our obsession with pasteurization. Thank you, Pasteur. He falsified the theory of spontaneous generation and verified that cells arise from only pre-existing cells, which then became a part of the cell theory.